Good morning, everyone. So today we assembled here to conduct the final viva of Junaid, Mr. Junaid, who worked under the supervision of Dr. Priyanka and Dr. Mudassir. I am pleased to inform you that we have among us Professor Reddy, who is head of the Department of Biotechnology for Kappa Institute. He has conducted his PhD research in the University of Madras and went to US and France for his postdoc. He has a number of publications, worked under different these project committees, including DBT, DST, ICMR, UGC. He supervised 31 PhD scholars under his belt, and he's a member of so many committees. He has awarded some prestigious award in his credit. So now we welcome you, sir, and with your permission, we will proceed with the proceedings. Yeah, please. Okay. Starting now. Thank you very much, everyone.
Good morning, everyone. Thank you for being here today. I am Junaid Nazir. I am here to present my work. I'm here to present my PhD work titled Development of Lactobacillus-based Recombinant Vaccine Against Salmonella in Poultry. This work was supervised by Dr. Priyanka and Dr. Saeed Mudassir Andrabi. And today in this presentation I will try to tell why Salmonella is a big burden in poultry as well as public health, and how we try to achieve this novel vaccine that we try to develop, and how efficient it is in the poultry farm settings. So Salmonella is one of the major zoonotic disease. It's not a problem only for the poultry, it's also a big burden in the public health also. So it continues to be one of the major diseases, accounting for around 150,000 deaths annually and 95 million Salmonella hospitalizations globally. So in poultry also, when we see the poultry is one of the major reservoirs for this Salmonella that is transferred to humans after consumption of the contaminated poultry products. So globally, it was the estimation was done around 20% of the poultry products are contaminated with Salmonella. And also it is one of the reasons why $3 billion losses happens every year to poultry industry because of the Salmonella. So the problem is that although there are some of the vaccines that are in the market, but we have seen that we even got some cases where the poultry farmers actually gave vaccines to the poultry, but still they are infected with Salmonella gallinarum, and that caused huge number of deaths in the poultry farms. And then when we saw what was the reason behind that, it was because the vaccine that they have used was serotype specific, and that because that gave the immunity against a specific serovirus, not around all the four major serotypes that are causing problems in the poultry. So we wanted to make a vaccine that will be multiple serotypes will be neutralized, because the vaccine will get immunity in the poultry that will be for all the four major serotypes. That's typhimurium, that's enteritis. These two are the ones, those are zoonotic in nature, and then there is gallinarum and chlorum. So those are a fowl typhoid that's causing problem in the poultry. So we will have we trying to make a vaccine that will have that will cause the immunity against all the four serotypes. And because of this, the vaccine that we are creating, so it will have both cell-mediated as well as the humoral response, and that was one of the targets for making the vaccine. So what kind of a solution we came up with was development of a vaccine that will have both B cell as well as T cell response in the poultry, and for that we used multiple tools to analyze, to predict the epitopes that are B cell epitopes or T cell epitopes in these antigens, whatever antigens we, and it was a kind of multiple antigen vaccine that we are going to make, and that vaccine will make a plasmid. It will go into the lactobacillus being one of the probiotics, that is plant derived probiotic that's already present in poultry, and it has good gut colonization impact. So we thought it's really a good vector for us. We can use this as a vector. It will go into the intestines and it will follow the natural route for the salmonella, like salmonella goes into the intestines and infects the poultry there. So we thought we'll use this lactobacillus plantarum. It will have these antigens anchored on the surface. It will go in the intestines, it will get colonized, and the immune system will come and interact with this lactobacillus plantarum, that is a recombinant one, and we will have immune response against the antigens that are on the surface of lactobacillus. So these are the objectives that we planned. So the first was that the bioinformatic analysis we will do, and we will do the research, we will do the literature review. So that literature review will tell us what are the different proteins that have been used already, what are the different proteins in the salmonella that are used, what are the different proteins which are conserved and which we can have in our vaccine, which are a good target for the salmonella protection. And then we will construct this plasmid that will go into the lactobacillus, and in the lactobacillus these proteins will be expressed on the surface, and then we can check the expression of these recombinant proteins that we have, and after that we will validate the vaccines in the poultry so that we can see how much efficacy we will get because of this vaccine. We will check the both antibody titer, we will check the cell-mediated response. So for that we will see the CD3 subset, CD8, CD4. We will see those subsets, what kind of growth, what kind of population growth we will get in CD8 as well as CD4. And for these we followed this methodology well. For the literature, we took multiple these studies that have been already done. So there are some of these antigens that have been identified. They are causing some immune effects in the poultry that have been already studied. So we took all those, and then we ran some bioinformatics tools, so that we will see which of these antigens, whatever antigens we have finalized. So we will see what are the 3 top antigens in them, so that we can use those antigens in for the salmonella vaccine. After that, we use this plasmid, that is PTRK892. PTRK892 is already found in Lactobacillus plantarum. So PTRK892 has very good PGM promoter, that's a constitutive promoter, so it will continuously make these recombinant proteins. And we also use a signal sequence so that these proteins will go outside the lactobacillus when they are expressed. And after that, we also used an anchor so that it will get attached to the surface of these this lactobacillus. It will get attached to it and it will be displayed on the surface of lactobacillus. So we also use the BSTX1 and NOT1 sites to make this cassette, expression cassette, and insert it in the PTRK892. And the objective third that we had is expression of recombinant protein. So after the transfection of this plasmid, recombinant plasmid in this Lactobacillus plantarum, we had to confirm that if the protein that we are targeting is getting expressed or not. So for that, we already use a His tag, in these proteins, so we had a his tag attached to it. So for the confirmation purpose, we also use the Sanger sequencing to see if our this construct is good and it has a good sequence for that. And we also use this restriction enzyme digestion also. So we use NOT1 and we used the BST-XR for restriction digestion, and then we checked the different fragments that we got, and so that we can confirm that our this construct is good. After that, we did the validation of vaccines in the poultry. So for that, we have taken 96-day-old birds, and we have used those birds, and we have inoculated this vaccine in the test group, and we have also used a control group so that we can compare the results. We have given a prime dose on day 1, and on day 10 we have given a booster dose, and after that 21 days, 24th day we used Salmonella gallinarum ID50. So infection dose 50 we used, and we gave it to the poultry so that we will see how many these birds will survive this ID50 dose. And we also did the post-mortem after 10 days after giving the challenge, so that we can see what is the condition of the organs inside these birds which have died or which have survived. So we also did the evaluation of antibodies, where we did the IgA as well as IgG. So IgA we use for the mucosal antibodies, because that is one of the major antibody that we will be requiring in order to get the protection from Salmonella in the poultry. So we did the ELISA for that. We coated the plates and with the antigens that we already used, and then we used the serum and this mucus of these chicken, and we saw how much antibody titer we will get for that. And we also did the B and T cell, this T cell, these T cell count. We saw CD8 and CD4. We saw how much it grows, the population grows after immunization. We use FACS for that, and we use ELISpot for looking at the cytokine expression. We saw that interferon gamma, how much interferon gamma is treated, because that will tell us how many Th1 or T helper cells or cytokine cells are getting activated because of our vaccine. So this ELISpot we did for that. And these are the results that we got. So in the literature, we got many these antigens that we found are good and potent, but SOPE2, PAGN, and FLIC. So two of these proteins are the surface proteins, but one more protein is the secretory protein. So what we have seen that most of the T cell response that we can see is generated more, the more skews towards T cell response because of the secretory, these antigens. So we have taken one secretory antigen, and two we have taken the surface antigen, so that will balance the T cell and B cell response in the vaccine. So when we use some bioinformatic tools, and we saw that there are a good number of B cell epitopes and T cell epitopes, MHC1 as well as MHC2 epitopes that are present in these antigens. So we used these ELIPRO, we used MHC-PAN tools for that. And after that, we went for the construction of this plasmid. So for the construction, what we thought is that we will first do the codon optimization. So codon optimization was one of the major things that we did. So for the after the codon optimization, we saw that we will synthesize these this expression cassette we got synthesized. So we synthesized it with the GenScript. GenScript synthesized this, and we have these antigens in expression cast. This expression cast we cloned in the vector that we already had PTRK892. So that expression cast and this plasmid made a recombinant plasmid which is ready to express the proteins, our recombinant proteins in the lactobacillus. So this is after the expression, how we validated it is, first we did the restriction digestion, and in the restriction digestion, if we can see that 6.6 kb, this was the original size of our construct. It is single digested, so we digested it in one place only, and after double digestion, we can see that we have this expression cascade different, and the backbone plasmid is 4.5 kb. And after that, we also did the Sanger sequencing. We saw that there is no mutation in the plasmid that we have inserted in the lactobacillus plantarum. So we also checked for the expressed protein, the protein that we got expressed, and we saw that in Western blot. We got that using the anti-His antibodies. We saw that we have this recombinant protein getting expressed. After that, we went for the validation. So the for validation, what we did that we administered these doses in 2 doses. One was the prime dose that was a day-old bird only. On the day 1, we inoculated that's an oral vaccine. So orally we inoculated this vaccine in the day-old birds, and after 10 days on day 10, we did the boost dose, and then we waited for the 14 days, which is the protocols for this. So we wait for that much time, and after that we went for the challenge. So in the challenge, so what we saw that around 94% of the birds that were inoculated with the recombinant lactobacillus plantarum, 94% of the birds, those survived, and only 56% of the birds survived in the control. Although this is not just a control, we have inoculated natural lactobacillus plantarum, and natural lactobacillus plantarum also acts as a probiotic. So that effect if we see, and that's why we got this huge number of survivors, 56%. Otherwise, this wouldn't have been possible, because immunomodulatory effects of natural NCAT is also there. So that's why we got this. And saw the serum, in the serum and in the mucous intestinal wall of these birds, we have seen that there is a good shift of this neutralizing antibodies, the binding antibodies that we have seen in the ELISA. We saw that there's a good number of good increase after the second booster. So this is the day 9 when we saw that after the first booster. So this was the titer, but it grew very huge on day. We also did it for the long term also. So we actually saw that after 70 days also. So in the ELISA, we have kept the birds for 70 days also in a separate experiment, and we saw that even after 70 days, the antibody titers were very high in the immunized birds, whereas in control it was less. So for the T cell assessment, what we did that we used the CD3 to select a population, and after that we used CD4 and CD8 to see how much increase in the population of CD8 and CD4 was there, and it was very prominent that CD8 actually grew very huge. I mean, it was a huge increase in the population of CD8, and CD4 was also increased, but for CD8 actually we talked to many scientists who are working with salmonella antibodies and so salmonella immune response. So the immunologists, we talked to them for this increase in the CD8, because we were not expecting this huge increase in the CD8. And then they said that for the pathogens that will internalize and stay in the dormant phase, which is one of the key features of Salmonella. So CD8 is one of the major factor how these pathogens are getting removed from the system. In the systemic infection also, CD8 plays a very huge role in clearing the infection. We also did the L-spot and we saw that the count of these dots which represent the interferon gamma excretion by the splenocytes. So we took the splenocytes and we saw that how this interferon gamma is getting excreted with the different peptide pool as well as a pool together when all these antigens were pooled together. So we got a huge number. But in these three also we got a very good number in PAG, and so we thought PAG and what we already expected that it will have a skew towards the T cell immune response. So we got that here. After that, we did the histopathology also of the birds that died and because of the infection, and the birds that survived in the infection, the immunized birds. So we saw that in the liver, intestines, or the heart. We saw that in all these tissues. So because of the systemic infection, Salmonella, this happened in the intestinal villi. So there was damage in the tissue. In these intestines and liver, and also there was congestion in this heart, these arteries. We could see that in the histopathology. And after that, what we did, so this was a good, whatever results we were getting, these were very good results, but there was one of the problems that we still had in our mind, that the eggs that are getting produced, because some of these birds that survive, they are becoming the carriers of Salmonella. Maybe they won't have any infection or any problems with them, but they transmit it through feces or through eggs also. So there are eggs also which are getting contaminated with the Salmonella. So one of the major problems with the Indian poultry industry is that there's a huge market, huge poultry, huge egg production, but we cannot supply these eggs to United States or some of the European countries which are looking for the Salmonella-free these certificates for that. So we thought we will see that how this vaccine is doing with the eggs. So we found that we did the experiment on the layers, and the layers also we did the one prime and one booster, and after that we did the challenge. In the control group, we saw that the eggs that were laid were having Salmonella, and then we could isolate Salmonella from them. And while as in the immunized birds, it was totally Salmonella free. So this was one of the things that we could. That's why we thought it's not only protecting, it's also stopping the transmission of this vertical transmission of Salmonella. Then we went for the mass trials where we saw how effective and how safe this vaccine is. So we went to the 5 farms where around 5,000 birds were in each farm. So we used our vaccine to do the mass immunization. In poultry farms, what they do is that they keep small herds of these chicks in one side, and then they take this vaccine in the pot, and chicks themselves drink it. So it's more easy to vaccinate or immunize the birds in that way. So this is the procedure that they are doing with the Ranikhet vaccine or the other vaccines that are oral vaccines. So we use the same strategy. We did that, and we checked the antibody titers. We took some of the birds, randomized birds we took, and first of all, we saw that there is no adverse effect because of this vaccine happening in the poultry. or there is no problem that's happening. And we also saw that in the randomized this bird selection, we saw that antibody titers were very high in the immunized birds. So why our vaccine is in the market also, it feels like our vaccine will be more ready to take, or more a successful vaccine, because the vaccines that are already in the market are serotype specific vaccines, or the cold chain is required for exportation and keeping the vaccine. So our vaccine is, it's one of the things is that it's probiotic, totally safe, no chance of getting virulent, like the inactivated vaccines which already have this problem. So, and also our vaccine, the dose per dose, it will be around per 100 dose, it will be around 50 rupees only, as compared to the other market, these competitors which have huge difference in the. This is the ethical clearance that we got from this cost, and this is from the DBT we got that the vaccine that we have made is safe and we can use it. So these are the references that we use while doing over this, and these are the publications that we published during this tenure. So some of the vaccines, some of the publications are still in the line. So this is one of the conferences that I attended, and I got best post presentation for this. So this is from the academy, this veterinary society, Indian Veterinary Society. They awarded me the best post presentation, and also we got Albert Lingard Memorial Award for making this vaccine. This is one of the vaccines that we made. For that, this award was awarded to us. And this is a letter of appreciation from Vice Chancellor for making this vaccine. And this is the patent that we patented for this vaccine. We got patented, and after patenting, we also got it licensed to one of the companies, Invarti Creations, because many of the companies were talking to us that they want to commercialize the vaccine. So one of the companies that approached us is Invarti Creations, and we have licensed this vaccine to them. And I would like to thank my supervisors and panel members for being there for me and for their guidance, and also DST for the funding for this project, and also the facilities from the institute, LPU and SCOST K, and also the lab and staff members and all my friends and family for the support during this phase. Thank you.
Thank you, Junaid. It's a nice presentation.
Thank you.
I just wanted to have some queries. So, what are the different species of salmonella?
So different species.
Which causes salmonellosis in poultry?
It's Typhimurium, it's Entitidis, it's Gallinarum and Pleurum. They are now taken under the same roof of Gallinarum only.
It's been more than one species.
Yes,
Is that?
4 species.
So based on that, there may be different antigenic properties for each of them.
Yes.
So what was the base in selecting these three?
So these three antigens that
which organism you have selected?
Yes, because these are very conserved across these these two serotypes. It's very conserved. It's around 99.9% conservation across all the serotypes that are in the poultry. So that's why we use these antigens. Otherwise, there were some more antigens also which had more antigenic properties, but they were different from, they were not conserved, they were not so much conserved. That's why we didn't take them. We took only these three, those were very conserved.
What I mean to say that every species have its specificity to express an antigenic property.
Yes.
To cause a different sort of diseases in poultry.
Yes.
Because every organism causes a different sort of a symptoms and diseases.
Yes.
Is that true?
Yes, sir.
Okay. But when we are talking about that, so is this proteins of the three which you are putting a cassette, so are they common across?
Yes, sir, they are common across all three, and they are doing the same activity in all the three these serotypes. Whatever serotypes we have in the poultry, these antigens have the same property, and they are doing the same work for the
I understand that. What I want to ask is that if these are specific to all the organisms,
Yes, sir.
then when you talk about the different species,
Yes.
so every species is recognized for a particular disease which is caused by this organism with some specificity.
Yes, sir.
So in such consequences, how do you say that this particular antigen is, though it induces immunogenicity in organisms, it's okay. But the severity of the disease, as you know, that it
Yes, sir,
varies
it's-
from one, and every bacteria have a different antigenic surface, antigenic properties.
Yes, sir.
So suppose if you are infecting with one organism, one salmonella species,
We did challenge with sir all four these serotypes.
then what is the differences in that?
Yes.
So that's what I wanted to understand. Suppose you target one organism, maybe this is doing some X percentage of inhibition or whatever it is.
Yes, sir.
So what happens if you challenge with X, Y, Z?
Yes, sir.
Is it the same response or is the response different?
It's the same response, and actually the Gallinarum and Chlorum are known for killing the birds. I mean, all the flock is dead if the Gallinarum or the Chlorum is infects this poultry. So we used Gallinarum also. We used the ID50 of Gallinarum. Then we saw that how much percentage of our birds are getting rescued. So it was 94% of the birds were getting rescued in Gallinarum, in Typhimurium, also in Enteritis. So more than 94%. We also did so after this because for the commercializing, what we did, we gave this vaccine to Tanvas. So the bacterial vaccine center that we have in the Tanvas institute. So Indian Bacterial Vaccine Institute, we gave our vaccine to them, and we got it validated there in the SPF birds also. So in the SPF birds, they have seen that this vaccine is giving more than 90% protection.
Okay, second.
Yes.
So when I'm talking about the construct,
Yes.
can you show the slide? So is this the complete cDNA or is a partial? Or did you?
No, sir, it's a DNA. It's all DNA. It's a plasmid only. DNA plasmid.
No, it's a plasmid item. So under this plasmid, you have cloned these three genes.
Yes, sir.
So they are, it's like a operon.
Those are also, yes, sir. Those are also DNA because we got it synthesized. So they synthesized the double-stranded DNA.
Did you completely synthesize the DNA?
Yes, sir. We
What is
completely.
the size of each of the DNA?
So we synthesized around. So it comes around 2100 base pairs.
For each?
Huh?
Each gene?
No, sir. No, sir. The whole this expression cassette that we got synthesized, it was 2100.
No, what is the size of each of these genes?
Each of-
Like 13 genes, na?
Yes, sir.
So what was the size of each of the genes? So why did you
this
synthesize this?
We could have amplified it also, but we synthesized it because we did the codon optimization. Because this is a salmonella gene, but we are going to express it in lactobacillus. So for that we need a codon optimization. If we codon optimize it, then the expression will be more.
How does it differ from bacteria to bacteria? Is that much difference you find in codon?
Not that much difference, but there are some differences, sir, because when we saw that the triplet codons that are used in this.
It's also bacterial system.
Yeah, it's also a bacterial system.
Then I don't think that you expect these codon bias to such an extent.
But there was some codon bias.
That's not my question. The question is that, so you have X, Y, Z,
Yes, sir.
3 genes. So you made a cassette.
Yes.
Okay? Is it operand system?
It's on. We have a PGM promoter, and it's a single peptide promoter system. We have a PGM that's a constitutive promoter. That's already.
No, I understand. I don't talk about the promoter and vector.
It's a
You
same.
talk about the 3 genes.
Yeah,
How
it's a
do you
single-
align them, one after the other?
Yes, sir. One after the other, but it will be expressed as a single peptide. not multiple peptides.
You cannot
It will
fix
be-
a peptide, it's a complete protein.
but
Three proteins.
no, but it's kind of a single recombinant protein. It will
So
be.
then can you tell me what is their expression places? All of them are surface?
All of them are on the surface because we have one this anchor, and with the anchor all the three peptides are attached, and that anchor is attaching to the surface only. So we'll have a single one. And
Is this an extracellular protein?
yes, sir, this is extracellular proteins that we're using. There is only one protein, that's secretory protein, but that also goes outside. So we have three proteins now. One protein.
Here there is nothing, no, you have only one protein.
We have one protein. We have
You
kept
do not
also-
have a three protein.
We have furin cleavage sites also. We have taken these furin
Then
cleavage sites.
how do you say it's a secretion is a different, it is an extracellular. Secretion is just throwing out into the system.
Yes, sir.
Okay, outside the system.
It will.
One is that, okay, expressing or attaching of this whole peptide as or not peptide is a protein on the surface of the lactobacillus.
Lactobacillus.
Okay.
Yes, sir.
So, whether when you have such huge protein, so have you even studied about their three-dimensional structure and also their
We are targeting the linear epitopes. We are not targeting the
but how do
linear
you say
epitopes
it's a
also.
linear epitope?
Because it's
when it
linear.
is a protein,
Yes, sir.
so protein has to fold to form a three-dimensional structure, then only it becomes a functional protein.
Functional
no?
protein.
So then, what about that? So, it's not like a thread where your cells can attach or the ligand can attach so that you will have antibody interaction taking
Yes.
place. So that's not the thing, no? Expression of the epitope itself.
Yes, sir.
Because every protein have a different structure, okay, three-dimensional structure where it can express its protein.
Yes.
When you made a fusion protein, okay, so have you studied about the interaction of these three proteins one after the other when you are forming a three-dimensional structure?
So after it's getting expressed, it's also when it goes in the intestinal cells. So in the intestinal cells we have furins. So but in the
You are
prokaryotic,
not understanding the simple concept.
I'm understanding that.
It's a three-dimensional structure protein. Every protein
Has its own
three
structure.
different proteins infused together.
Yes, sir.
Is that correct?
Yes, sir.
So it is I don't know, I cannot also call it as a multimeric because multimeric protein in size will differ because to become into a functional protein by different subunits
Yes,
is
sir.
called as a multimeric. But here you have created a protein of fusing three different types of proteins. When you look into individual protein, they have a different three-dimensional structure,
Structures.
and when it is exposed to any surface or any other environment, so the epitopes, whatever it is, it has to express its antigens, where antibody can rise onto that. So if I say one thing, I can see. But when you are fusing three, what type of protein-protein interactions which it is taking place to get a structure? So where did you validate the structure of the protein?
So, sir, because we were not targeting.
You are not targeting about this, so how can you say that? But okay, for before that, did you have even estimated about the immunogenicity of the lactobacillus alone?
Yes, sir. The
What is the differences which you find? Whatever the parameters which you have studied, because it's a probiotic,
Yes.
it means that it also has the
it also has the immunomodulatory effects.
And what is the lactobacillus and salmonella interaction?
So in the only salmonella, natural salmonella that
I am talking about what is the interaction between salmonella and lactobacillus when you put them in a combinedly, when you put in a system. So what is there antagonistic or is there mutual or they don't have any interaction?
we haven't done that study, but that's
And you are
already
not
there.
doing it.
That study is already there, sir.
I'm not criticizing. When you are not doing that, what is the guarantee that whatever the immunogenic responses which you are getting, is it not from your lactobacillus?
alone.
alone.
So the control we have taken lactobacillus
How
only.
many fold increase you saw? When you compare to control as a lactobacillus, what is the fold increase of immunogenicity in your cells? What is the fold increase compared to lactobacillus?
Compared to lactobacillus,
Suppose lactobacillus is a probiotic. Okay?
Yes, sir.
So it means that whenever any probiotic, it also have an antagonistic or antibacterial property. It also have immunogenic properties because they also act as immunomodulators.
Yes.
And all these properties are there with that particular organism. Clear?
Yes.
So now, when first of all, I have this is my doubt. I am not doubting your credentials and all. Whenever you made a multimeric protein of such kind, which is not present in nature as such,
yes.
okay, and you combine this and form into a fusion protein. If I am in your place, I will study first their protein-protein interaction. Okay, because how does the structure, because sometimes the most immunogenic epitopes which are present in your peptide might be buried and not exposed to your the process. When you talk about a 3 different proteins,
Yes.
when I am talking one protein, it's already known, accordingly it folds so that the antibodies can bind and the immunogenic responses can be initiated. But when you are combining 3 different proteins, there isn't much interaction because of that there may be more foldings or more openings, I do not know because you have not studied. If that is the case, what guarantee that all the epitopes are exposed and doing their immunogenic responses in your system? So this is the first question. So for that, you should have done the protein-protein interactions and also you should have predicted the 3-dimensional structure of this protein by using the bioinformatics tools alone. Then you should have studied a docking. Okay, you see how the antibiotic or interact with your antibodies, though you estimated the titers of immunoglobulin G, that's not the thing. That's a different issue altogether. Because now you made some protein, maybe it's good, okay, maybe all are exposed, but you are not sure about these experiments what you have done it. And also, that's what I said, I need to know that what fold increase in the immunogenic properties you have observed when you vaccinated the plasmid associated bacteria and normal lactobacillus in the form of a probiotic.
So that is what we took in the controls. So the control we have taken normal lactobacillus.
What fold did you find?
So this is in the normal lactobacillus what we have taken in the control. So we got only 10⁻², this endpoint titer of these, but in this we got around 10⁻⁶. So we have done boosters of natural and lactobacillus plantarum in controls also. Even after doing the boosters of natural lactobacillus plantarum, because when we see the ELISA, so ELISA is against the particular antigens that we are. So we coat the plates with the particular antigen, particular proteins, and after that we use serum to see that how much of this antibodies are getting how much antibody can we see in those are against the particular antigens that we have coated on the ELISA plates. So in the natural lactobacillus plantarum, we didn't see any antibody titer against those antigens that we have in the recombinant vaccine, while as in the after the prime booster we had.
No, that I can understand because you don't have a plasmid here.
Yes, sir.
Okay, you have a plasmid in the other place. So there may be some response.
Yes, sir.
Maybe one or two epitopes might be exposed against that particular antigens. You might have an antibody titer.
Yes.
I can see. So what about the titer of T cells?
Titer of T cells. So we in the T cell also we used all 3 different antigens that we have separately, and we have the PA.
Separately, how can you do it?
Because
That is a protein.
no, then those peptides we get synthesized. Again, we synthesized those peptides. Then we coated these.
No, that's what I wanted to ask this question.
Yes, sir. Those are
One thing for beta, so like beta cell are
yes, sir, those are separate. We didn't use this recombinant protein.
Then what is the use of constructing this plasmid?
So the plasmid will be used in the probiotic in the vaccine construction.
But for whatever the studies like T cell development, B cell development, and also immunogenicity, all these things should go against these.
The epitopes of these, the predicted epitopes, whatever. So we have done the prediction of these epitopes, and we have seen that these are the different epitopes that we have seen that MHC1
सारे समझ गए I'm not telling. अगर आप इंडिविजुअली करेंगे, that's what I was asking you all this. इंडिविजुअली करेंगे तो आपको ये कोई भी जो प्रॉपर्टी है that will be induced or expressed.
So we are only seeing that our vaccine, whatever we have immunized the birds, in the birds, when we immunize the birds, so that vaccine able to generate the response against these peptides that we have already predicted. If the response is against these, that means these epitopes are getting expressed in the Lactobacillus plantarum. The vaccine that we have, that means that we have.
Now, how did you express this?
This.
What is the method you studied?
These ones, sir. These ones. We have synthesized these.
You have synthesized.
Yes.
I'm not telling that. So suppose if you say that there is one way of expression is that for T cell, for a B cell you can estimate the amount of antibody.
Yes, sir.
Okay, titers. Okay, then based on control, you say that there is this much fold increase in the immunity.
Yes, sir. For the T cells, we used FACS for the CD8 and CD3, and we also used ELISpot, so that we will see that how much activation is happening because of the peptides. When we keep the splenocytes, we use peptides to activate them. If they already have been exposed to these peptides, they will get activated and they will produce interferon gamma. If they are not getting activated, they will not produce any. So this is one of the negative controls that we have.
See, when you have synthesized these peptides, you tested these peptides against by challenging these by incorporating or injecting these peptides into your.
No sir. Not injecting. So we use this plate, the ELISpot plate. This is one of the plates.
That's in vitro study.
Yes, it's an in vitro study. These are all in vitro studies that we have done. We have only extracted the splenocytes from these immunized birds. So from the splenocytes, if the splenocytes are already have been exposed to these antigens or epitopes, or they already have T cell response they can generate against these peptides, only then they will get activated and they will make these spots. That's why we got spots only in the immunized one, not in the control bird. So in the control bird also we use the peptides, but there is no response.
Now we should test them in the eggs. Means that what is the method which you can say that these eggs are salmonella infected?
Okay, so we directly plated the eggs. We plated the egg white as well as the yolk of the eggs. We plated on these plates. Agar, yes, XLD agar or SS agar, which is very common for the salmonella this identification. So we used those, and we saw that in this egg, in this plate we have seen that these black these colonies in the XLD agar, which is very common for identification of the salmonella. So we saw that we could isolate salmonella from.
Did you check lactobacillus alone, lactobacillus with plasmid?
Yes, sir.
Against salmonella under in vitro?
Under in vitro. So under in
This is the microbial interaction between them.
No, sir, we didn't check that.
Actually, see, salmonella is gram-negative. Lactobacillus क्या है?
क्या सॉरी ग्राम पॉजिटिव है ना लैक्टोबैसिलस
जो नॉर्मली जैसे सर हाइपोथेटिकली जगह पे लैक्टोबैसिलस जब भी हम खाते हैं उसका जो भी एक्सक्रीशन होती है
उसमें
वो gram-negative के अगेंस्ट में एंटागोनिस्टिक है ही है. I checked it in case of Pseudomonas. Right? Pseudomonas and Salmonella very close to each other. Equilibrium है. So इसीलिए जब वो एंटीबायोटिक देता है तो आपको कहता है लैक्टोबैसिलस खाओ just to settle down कि उनका एंटीगोनिस्टिक इफेक्ट है. अब वो वाली स्टोरी जो सर कह रहे हैं ना, मतलब वो स्टोरी बहुत इंपॉर्टेंट स्टोरी है। मे बी सर मुझे लग रहा है कि ये जो है ना वो अगले स्टेप में जाएगा एंड समहाउ इनको ये चैलेंज आना है ड्यू टू देयर जर्नी ना, दे फाइंड दिस चैलेंज।
That's these three things are very important.
Yes, ये चैलेंज आएगा।
One is your protein interactions. Okay, because if you go to local companies, they may say because it's animals, so nobody cares.
Yes.
If it is humans, then you will be less screwed.
ये वही वाली स्टोरी है, तुम बाहर भी जाना ऐड होगा सारे क्वेश्चन।
See, all actually that because when we say that this advantage of having lactobacillus is that it's a probiotic. So whenever you say probiotic, it means that it will protect your system by itself.
Yes.
So I just wanted to know that what is the level of protection which you can get
only because of the this lactobacillus.
Simple as
In the
interaction. दूसरा आपका
in Salmos also they have made 3 groups. One group they have kept only PBS. The other group they have kept natural lactobacillus, and the other group they have kept the recombinant lactobacillus.
सर इंडक्शन कैसे होती थी? You just एक फ्लास्क में स्मॉल सालोमेला डाला, लैक्टोबैसिलस से 2 डाइल्यूशन में डाला and 1:10 और उसमें देखना था कि ग्रोथ से इंजेक्ट हो रहा है कंपेयर टू सालोमेला अलोन एंड लैक्टोबैसिलस को 4 मिनट का एक्सपेरिमेंट एंड यू विल गेट यस
full microbiology test.
वो होना हां वो यस।
अच्छा, when you are challenging the dose to the animal.
Yes, sir.
So what does the dose have any influence on the immunogenicity?
So if we give the dose, that also
What is the delivery system of these dose?
we give intraperitoneal. Intraperitoneal doses we give.
You inject it?
Yes.
Okay. Okay. Not through. So you inject bacteria?
Yes, sir. We inject bacteria in PBS.
And then how do you say it's a probiotic?
No, sir. This one we give orally only.
Yeah, that's what I.
Yes, sir.
When you are so in which form you are giving?
We are giving this lactobacillus. We are suspending it in PBS. Then we are giving orally.
In what? Where do you suspend?
In PBS or this saline. PBS or this no.
ये आप कैसे देते हैं? अगर चिकन का कोई पोल्ट्री फार्म में जाके अगर आपको वैक्सीनेशन करना है, which form you are giving the feed to these animals?
Oh, in sterile water they make these dilution.
You add the bacteria and you give in a sterile water. Okay.
Convert.
So when you have like 100 chicks, okay, you have given water. Suppose some of them take more water, some of them will take less water. Does the dose of these inoculum have any impact on the immunogenicity of the chicks?
Higher doses have not caused any problems. So because we went doses 10 times also.
You cannot hypothesize it.
We can.
Do you have it?
We have done this because this dose depend also we have done. That's why we used only 10 is for 8. Otherwise we would
ये मैंने आपका बात समझ गया, वो तो लैब किया। When I am going into the field, ना, I don't have a control.
उसमें सर वो the
Because for this, these experiments are lacking. Okay, so this is the most important thing.
Maybe
Because
नहीं बताओ हमें maybe R&D can be done by PI क्योंकि इसको तो एक पार्ट दिया गया है complete vaccine development इसके सामने हो गई maybe they have their study but not shown to him maybe
because that
कोई और बच्चा कर रहा होगा कोई PI
those optimization was also done.
ये जो मैंने न्यूज़ सुनी भी है this is very good 10 साल का काम बोल रहा
हाँ, sir.
extra years work what they are doing इसको तो 2-3 साल का काम दिया गया है whatever the experiment they will have in their mind
see, whatever you project, what I mean to say, by our doing all these things, the ultimate, whenever you show any slide, you own it. Okay, it means that you are showing this slide because you are the owner of it. So when I ask anything, you should tell about it, whether somebody else has done
Yes.
or not, it's not my business. Okay, whatever I speak, when I show anything, I own it. It means that if you know, then only you can show that slide. Otherwise, we don't show the slides.
So this dose optimization was also done. I mean,
No, I am not telling you that you are not doing it. That's a different issue. Because these are all the things which at least you should have known. Because these are the things, entry points where you can get a questions for this.
but किसी R&D कंपनी के पास जाओ ना ये सारे क्वेश्चन उन्होंने पूछने लोकल वेब तो ले लेंगे नहीं
Also,
पूछेगा वो सारे क्वेश्चन पूछेगा तो उसको भी चार्ट सेफ्टी डाटा इसी से बनेगा जो उसकी डाटा वाली सेफ्टी शीट्स बनी है डाटा शीट्स की 5000 वर्ड्स कितने बच्चे कितने मरे तो बहुत अगर वो उसी से बनेगा डाटा
anyway, I think I'll stop here.
good job
You can
did a good job you did a good job
the rest of the things, I think if you have anything else, you can.
now कुछ पेपर ड्रिल होनी साइन करवाना लो पेपर लो हां
It's a good thing.
clinical biochemistry.
It's a clinical biochemistry.
It is okay. Whatever it is. Because the things which are essential is that these are the things when one should know.
And I think a part of their work, when they have students, and I think most of them just get nervous.
Yeah.
So I think you have to conduct one another viva.
That is after me.
That is after.
Yeah.
But I think they are in a mood that they should continue.
I will be more than happy so that I can leave my place. Are they ready?